This video demonstrates a method for measuring bacterial load and generating and analysing single-cell suspensions from mouse liver and spleen. First, a Listeria inoculum is prepared and injected intravenously into mice. Blood, liver and spleen are then harvested from euthanized mice at chosen time points. Single-cell suspensions are prepared from harvested tissues, and tissue homogenates are cultured on horse blood agar plates. The resulting cell suspensions can then be analysed for viability and immune cell content by FACS analysis, while the bacterial load is determined based on colony-forming units.
This method can help characterizing immune responses to Listeria infection, such as those between susceptible and resistant mouse strains, while simultaneously determining bacterial load in infected organs.
Begin this procedure by preparing the Listeria inoculum to the desired concentration. Instructions for culturing, storing, and preparing Listeria can be found in the accompanying written protocol. Once the inoculum is ready, transfer 2 aliquots of 100 microliters to sterile microfuge tubes containing 900 microliters of PBS to make a 1:10 dilution. Then, transfer 100 microliters of each dilution to horse blood agar (HBA) plates and spread thoroughly with a sterile spreader for performing colony-forming unit (CFU) counts. Then, incubate the plates, inverted, at 37 degrees Celsius overnight. Next, transfer the mice to a clean cage. Place the cage 50 cm under a 250 watt infrared lamp for 5 minutes to ensure that the tail vein is dilated. This allows for better visualization during the injection. Here, the Listeria-resistant mouse strain C57 Black 6 and the Listeria-susceptible mouse strain Balb C will be injected. Gently mix the Listeria suspension by inverting the tube a few times. Then load a 1 ml syringe with a 27 gauge needle for injection of 200 microliters per mouse. Ensure that any air bubbles within the syringe are bled out prior to injection. Place the mouse in a restraining device. Then, locate the lateral tail vein of the mouse by first identifying the tail artery, which is found in the middle of the dorsal side. Then turn the tail slightly to the lateral side to identify the vein. Gently wipe the tail with a 70% ethanol wipe. Then, inject. After withdrawing the syringe, briefly apply pressure to the entry wound with a sterile gauze square. Note that it's much more difficult to identify the tail vein in black-coloured mice. Following the injection, return the mouse to its cage. After injecting all of the mice, plate and incubate the remaining inoculum for colony-forming unit counts as before. The next day, count the number of colonies and determine the pre-injection and post-injection concentrations of the Listeria inoculum. The characteristic halo surrounding individual colonies, as shown here, is due to beta hemolysis. Once the pre- and post-injection counts have been determined, calculate the average colony-forming units by adding the pre- and post-injection colony-forming unit counts and dividing by 2. Finally, at various time points following injection, collect blood and harvest the liver and spleen from euthanized mice. If FACS analysis will be performed, the liver must be perfused before removal. To obtain blood, perform a cardiac bleed immediately after euthanasia and collect as much blood as possible in an appropriate collection tube. To perfuse the liver, first open the abdominal cavity and remove and discard the gallbladder. The anterior vena cava is a vein that runs parallel to the spine and transports blood from the lower part of the body, including the liver, into the heart. Using a pair of scissors, sever the vein just below the heart to create an outlet for the perfusing fluid to exit. Next, flip the liver lobes towards the head and push the intestines to the right to expose the hepatic portal vein, which feeds into the bottom of the liver from the lower right-hand side. Insert a bent, half-inch length, 26-gauge needle in the hepatic portal vein and inject 10 ml of PBS. After 1 to 2 ml of PBS has been injected, the liver will turn from a dark red colour to light brown. The injected solution will exit from the severed inferior vena cava. Liver perfusion ensures that harvested cells are from the liver and not the circulating blood. Next, remove the perfused liver from the mouse's body cavity and submerge it in a tube of Fax buffer on ice. Then, remove the spleen and place it on ice for processing in a similar manner as described in the next section for the liver. To generate single hepatic cell suspensions, begin by transferring the perfused liver from Fax buffer to a 70 micron cell strainer placed inside a 60 mm petri dish. Using a 5 ml syringe plunger, push the tissue through the cell strainer into the petri dish. Transfer the single cell suspension to a 50 ml conical screw cap tube and bring the total volume to 40 ml with cold Fax buffer. Vortex the cell suspension. Then, set a 1 ml aliquot aside for determination of bacterial load later in the procedure. Place the 50 ml tube in the centrifuge and pellet the cells by spinning at 500 times G for 5 minutes at 4 degrees Celsius. After discarding the supernatant, repeat this wash step by resuspending the pellet in 40 ml of cold Fax buffer and centrifuging again. Resuspend the pellet in 20 ml of isotonic percol at room temperature. Centrifuge the cell suspension at 700 times G for 12 minutes at room temperature. Following the spin, a disc-like sheet of hepatocytes will float on top of the percol and the leukocytes will be pelleted at the bottom of the tube. Discard the hepatocyte layer and percol by pouring it into a waste container. Then, resuspend the leukocyte-containing pellet in 4 ml of TAC buffer to lyse erythrocytes and incubate the cells at room temperature for up to 10 minutes with frequent inversion. Following the incubation, filter the cell suspension through a 100 micron nylon membrane into a new 10 ml centrifuge tube. Underlay the filtered cell suspension with 1 ml of Fax EDTA buffer as shown here. Then, centrifuge at 350 times G for 5 minutes at 4 degrees Celsius. After the spin, resuspend in 5 ml Fax EDTA buffer. Then, spin again and resuspend the pellet in 0.5 to 3 ml of Fax EDTA buffer depending on the size of the pellet. Determine the number of viable hepatic leukocytes using a hemocytometer based on trypan blue exclusion. Liver single-cell suspensions can then be labelled with antibodies specific for desired cell surface markers and analysed by Fax. Once single-cell suspensions have been generated from liver and spleen, tissue bacterial loads will be determined. For part of the procedure, a stomacher machine will be used to homogenize the tissue. Holding a stomacher bag shut to prevent leakage, begin by laying it flat on a clean bench. Roll a thick round pen over the tissue until the tissue is mashed within the bag. Add 5 ml of PBS to each stomacher bag containing mashed tissue. Place the stomacher bags into the stomacher and run on high speed for 10 minutes. The stomacher machine has 2 paddles, which press the stomacher bags at a specified speed, blending the contents into a uniform homogenate and containing any aerosols. Using a pipette, mix the homogenate in the bag. Then, transfer 300 microliters in duplicate to a 96 well flat bottom plate. Within the wells of the 96 well plate, perform 1 to 10 serial dilutions to a 10 to the 5 dilution for each tissue homogenate sample. Carefully, use a sterile spreader to spread 0.1 milliliters of each dilution onto a pre-dried HBA plate. Incubate plates at 37 degrees Celsius overnight. The next day, count the number of colony-forming units for each dilution and calculate the colony-forming units per tissue, taking into account the portion of the tissue, dilution factors, and volume of homogenate. C57 Black 6 mice were infected with around 2,000 colony-forming units of Listeria. At 3 and 7 days post-infection, mice were euthanized and the liver was perfused and harvested with the spleen. Dilutions of splenic homogenate or hepatic single-cell suspension were cultured on HBA plates to determine the bacterial load. In the results shown here, the dotted line indicates detection limit at 100 colony-forming units per organ. Note that the Listeria load is higher at day 3 than day 7. This is because by day 7, various immune cells lead to the clearance of Listeria in these two tissues. Spleen and liver single-cell suspensions were stained with trypan blue and loaded onto a hemocytometer. Cells within the counting area, indicated in the red box, will be counted. Non-viable cells will take up trypan blue and appear blue, whereas viable cells will remain unstained. As shown here, the number of viable cells in the spleen, shown on the left, and the number of viable leukocytes in the liver, shown on the right, increases during the course of infection. This increase is due to immune cells, including neutrophils and lymphocytes, that become activated, proliferate, and migrate to these two sites of infection. As an example of immune cell levels during the course of Listeria infection, representative FACS plots and the number of CD8 positive T cells are shown here. The number of CD8 positive T cells transiently decreases due to lymphopenia in the spleen at days 2 to 3, before increasing noticeably from day 5 post-infection in both the spleen and liver.
Following this procedure, other methods, such as isolation of antigen-specific T cells and functional assays of sorted immune cells, can be performed to further characterize immune responses to Listeria infection.